Hey guys, so in this one we are going to be talking about the esophageal slice of the GIT. So I'll cover the entire GIT in this one. So let's quickly get into it. Firstly, we need to remember that any GIT structure will have four basic layers. Starting from inside to outwards, first we'll have the mucosal layer, which will consist of the epithelium, the connective tissue that supports the epithelium, and then we'll have the muscularis mucosae. Then moving ahead, we'll have the submucosa. The submucosa will usually contain adipocytes or some blood vessels or some glands in this case, in case of esophagus, not everywhere. Then we'll have the muscularis externa, which will have the inner circular and the outer longitudinal. Mostly, except for stomach, there will be stomach and colon, there will be some modifications in this layer, which will be a significant feature. Then we'll have the serosa or the adventitia outermost. So these are the basic layers for any GIT structure. Now starting with the esophagus, I'm trying to focus on all the key features. So once we know the key features, the other features are more or less the same. So the first key feature of esophagus is its stratified squamous epithelium. So as we can see over here clearly, that we can see there are multiple cell layers at first. Then we'll have this uppermost flattened cell layer, which is the squamous tissue. So what happens is this becomes the stratified squamous epithelium. Then the second important feature in case of esophagus will be the mucus acini of esophageal glands, which will be present in the submucosa. So an MCQ had been asked, so where is it present? It is present in the submucosa, not the mucosal connective tissue. Okay. Moving ahead, we'll have the muscularis externa, we'll have the inner circular layer and the longitudinal layer, and then we'll have the serosa. This I have marked this as 1. So this 1 is serosa. Okay. So moving ahead to the stomach fundus. So what I'm going to try to do is I'll explain the stomach fundus, and whatever the features I'll then explain the stomach pyrolus as a distinguish between these two. Because there are many similarities and quite a few differences as well. So I'll explain the differences, the rest will remain the same. So firstly, the epithelium is the columnar epithelium. Okay. Then we can see these gastric pits which are present. These mucosal folds have taken place and there are some gastric pits which are present over here. So these gastric pits are not that deep in case of fundus. And at the end of these gastric pits, we'll have these glands. These glands have these basophilic cells and these eosinophilic cells. So the eosinophilic cells in these gastric glands are the parietal cells, and the basophilic cells are the peptic cells. So we know very commonly that the parietal secretion will consist of HCl and the Cassel's intrinsic factor. So the Cassel's intrinsic factor is necessary for the absorption of B12. Then substances like pepsinogen will be secreted by the peptic cells. So these are the secretions, right? But what we need to remember all in all is that a gastric glands is a very important feature of the stomach fundus. Okay, so this is a very prominent feature and very important feature. The next feature that we can use to differentiate basically stomach from any other structure will be the oblique muscle layer. So the oblique muscle layer will be inner to the circular layer in this case, and it is a very important distinguishing feature. Moving ahead to the stomach pyrolus, as I said, the distinguishing features between the fundus and the pyrolus will be that the gastric pits are deeper in this case, as you can see clearly. So the pits are way deeper, and the pyrolic glands are present instead of the parietal and peptic cells, right? Here we can see parietal and peptic cells. Here we cannot see the parietal or peptic cells. So the pyrolic glands will be present. The rest of the features will be similar to the fundus. Moving ahead, the duodenum. The duodenum will have a variety of features are present. Firstly, we can see that the mucosal foldings have led to the formation of these villi. These multiple villi have been formed. This is the muscularis mucosae layer, which I am showing. And then these folds are the various villi. on which we have this epithelium which has microvilli in it. So what happens? We have got one fold, we have got two fold, then microvilli have their own folds. So what is happening? The surface area is increasing many folds, approximately 200 folds, and that is why this plays a very important role in digestion as well as absorption. Okay, moving ahead, the most important feature of duodenum will be the submucosal Brunner's gland. Submucosal Brunner's gland. So these will be present in the submucosa. Here we can see a lymph node right here. Now, so what these glands, whatever the secretion of the duodenal glands will be, Brunner's gland will be, they will secrete their secretions into the lumen via the crypts of Lieberkühn. So here will be the opening of the crypts of the glands at the crypts of Lieberkühn. Okay. Then moving ahead, we have got the normal inner circular layer, the myenteric plexus which is present in between the two layers, the outer longitudinal layer right here, and then this is the serosa with some blood vessels. So duodenum is done. Moving ahead to the jejunum. So now the feature that we'll use to describe jejunum as a separate entity will be the Paneth cells. Okay. So all in all, the structure remains the same. We can see the villi are present, we can see the epithelium is present, then the microvilli will be present on the epithelium, different types of various goblet cells will be present which will secrete mucin type 1, type 2. So like these are the basic general features which were similar to the duodenum, but the differentiating feature is that instead of a submucosal gland, Brunner's gland, we'll have mucosal intestinal glands or mucosal jejunal glands, which will consist of pyramidal cells which are called as Paneth cells. Okay, so this is the feature, that is like the differentiating feature. Also we'll have the lacteals which are present over here in the submucosa. So what the lacteals will do is they'll basically help in the absorption of fat, and then the blood vessels, the inner circular layer, the myenteric layer, the outer longitudinal layer, and the serosal layer with some adipocytes. Okay, done. Moving ahead. Okay, this is the ileum right here. So now the ileum will have more goblet cells. Now this is one of the features that we need to remember, because if a question had been asked like the maximum number of goblet cells or like increasing order of goblet cells is seen, right? And the second feature, the second important feature will be like basically the differentiating feature will be the presence of these lymph nodes. These lymph nodes arranged at intervals which is present just adjacent to the muscularis mucosae. We can see this is the muscularis mucosae, and just adjacent to that we can see these are these lymph nodes which are the Peyer's patches. Okay, so these Peyer's patches are present in case of ileum, which are basically lymph node aggregations. And the interesting thing, the clinical aspect of this is that whenever peptic ulcers usually occur, they will occur in case of like in the region of ileum. Now and as oval patches. Now why is that, right? So that the answer is Peyer's patches, because these like oval patches will be seen in case of ileum. So whenever peptic ulcers occur, they will occur as oval aggregations, which will be because of the presence of Peyer's patches. So that is one interesting clinical aspect. Then the rest of the layers are pretty much the same. We'll see the lacteals are present, we'll see the inner circular myenteric plexus, we'll have the outer longitudinal, and we'll have the serosa. So we are done with ileum as well. Moving ahead, the final structure of GIT in this case will be the colon or the large intestine. So the colon, like the most important feature will be that villi are absent. This I've like shown this as villi is absent, but remember that villi are absent. These are just the mucosal folds. These are not villi. Secondly, and the most important feature, like if you are drawing colon, how will you draw a colon? like, because there is pretty much like the same thing. You'll feel like ileum was the same, jejunum was the same. So I'm mentioning the key features. Over here, the key feature is this outer longitudinal muscle layer is in just one region, arranged in one region. This region and this arrangement of the outer longitudinal layer in one region is called as tinea coli. So there are 3 such tinea coli present in colon, and if they ask you in viva that why are hostrations and saculations seen in case of colon, so the reason is that the tinea coli, or the layer of longitudinal muscle, is smaller than the mucosa. Okay, so it's smaller than the other layers basically. So what happens is you need to adjust the other layers with this length. So let's say this is the layer of the muscularis mucosae, and this is the mucosal layer, right? So what happens is this is smaller than this. So what will happen is you'll have to fold this over here to fit the entire mucosal layer onto the tinea coli. So that is why the hostrations and saculations are seen, and we are done with the intestine. So these are all the various histological slides that will come across in case of the intestine. So you can take a screenshot right here and right here, and you can prepare well for the histological slides. That's it for today, guys. I hope you enjoyed the video, and if you did, share this with anybody who is facing their practicals or who is going to face their practicals.